I'm Jennifer Smith. I am the Scientific Project Manager for the Rat Genome Database and the Variant Working Group Lead for the Alliance of Genome Resources. And today we're going to be exploring variant data at the Alliance. So today we're going to cover searching for variants at the Alliance, the alleles and variants section of the Alliance gene page, the alleles and variants details table, the allele variant page that gives more information about an allele or a variant, and then take a look at variants in the Alliance JBrowse genome browser, and we'll give you a sneak peek at the Alliance PAVI tool, and then we'll have a little bit of time for questions and answers. So one easy way of getting an overview of everything that the Alliance has is actually to do an empty search. So this is the front page of the Alliance website, and you can enter data or enter something into this keyword search, but you can actually not enter anything and just click this search icon, and you will get a summary of pretty much everything that we have. But to start off with, I want to clarify what we mean when we use the terms allele and variant, just so that we're all on the same page when we're talking about this. So at the Alliance, an allele is one of multiple possible forms of a functional genomic element. So usually that is a locus or a gene that differs from the reference. And a variant is a DNA sequence that differs relative to a designated reference sequence. So in the case of all of the alleles that we'll be talking about today, they are all actually altered genes. And then the variants that we'll be talking about are DNA changes that are associated with one of those genes or overlap one of those genes. So in this summary, if you choose the allele variant category, you get a number of options for ways of filtering that data, because you probably don't want to look at 43 million records. So you can choose a species. So I just want to see the mouse variants, or I just want to see the C. elegans variants. Or you can choose a category of alleles or variants. You can filter it by variant type, so a SNP or an insertion, or you can filter it by molecular consequence, a missense variant, a frameshift variant. And then there are additional facets that I'm not showing here that limit the results by disease, by gene, or by transgenic construct component. So one of the reasons that I wanted to point this out was because we do have human variants, and you'll notice that it's not every variant that has ever been seen in human, but we do have the clinically relevant variants from NCBI's ClinVar database. So it's a data set of almost 3 million variants that have been predicted to be clinically relevant. So you probably don't actually want to look at everything in the Alliance. So we're going to start by talking about some ways to find variants at the Alliance. So there are a number of ways that you can do that. You can search for a gene and then go to the alleles and variants section of the gene report page. You can search for an allele symbol and navigate to the genomic variants information section of the page. Or you can search for a variant directly using HGVS nomenclature or an RSID. I'm only going to talk about the first of these options in any detail. So if you want to search for a gene, you can put it into this search box on the Alliance front page. So I want to look for the leptin receptor gene, and because I work for the Rat Genome Database, I'm interested in the rat LEPR gene. So if I click this in the drop-down list, it actually takes me directly to the top of the LEPR gene page. And if I'm interested in alleles and variants, I can just scroll down to the alleles and variants section of the page, or I can use this link in the left sidebar that will take me directly down to that section. So if I click that, that alleles and variants link, it takes me down to this section. This is the top of the alleles and variants section. And I want to point out first that we have a number of these question mark icons that if you click it, it will open up an informational pop-up with context-specific information. In this case, it will give you the definitions of alleles and variants. The next line in this section is a link to JBrowse for the genome location of this gene. And if you click this, it will take you directly to the JBrowse, Alliance JBrowse, to see this gene and all of its overlapping variants in their genomic context. And I'll be talking about this again a little later in this presentation. So right below that, there's a sequence viewer that shows you the allele-associated variant. So that's these two icons right here in the context of the transcripts of the focus gene. So LEPR, that's the gene page that we're on. So that's our focus gene, and also transcripts of any overlapping genes that are in the area. So here we see that it has the LEPR overlapping transcript gene is not surprisingly in the same region and is shown in this view. So below the sequence viewer, there is a table of all of the alleles and variants that overlap this gene. So the first column contains the symbol of the allele or the variant. The second column is any synonyms that we have for those. So if you know the allele by a different name, you should be able to find it in this column. And then there is a column for the categories. So the first category is just called allele, and these are phenotypic alleles for which the genomic variant either isn't known or it hasn't been curated. So you'll notice that there is nothing in the information for variants that are in that row. So the second category that is shown is phenotypic alleles with one or more known genomic variants. In this case, for the rat LEPR, we have two alleles that have one associated variant each. If there's more than one variant for the allele, it will show all of the variants that are associated. So this is an example of a fly allele that has 3 variants associated with it. So the category is allele with multiple associated variants, and it gives you the information about all 3 of the variants. So if you click a row for a phenotypic allele that has one or more known genomic variants, it will highlight that allele in the table and also highlight that allele in, or highlight the variant in the sequence viewer display. So if we go back up to the sequence viewer, you can see that this variant is highlighted in yellow. And this is actually reciprocal. If I click on the variant in the sequence viewer, it highlights this variant as it's shown now, and it also will highlight that allele or variant in the table, so you can easily find where that is find the information for it in the table. And when you click the variant, it also will bring up an informational pop-up with information about that variant. So going back down to the table, the last category is just labeled variant, and these are genomic variants that are not known to be associated with a phenotypic change. So these are usually derived from large-scale sequencing projects like whole genome or whole exome sequencing. If an allele with or without a defined genomic variant has been associated with either a human disease or a phenotype, then this association is indicated in the last 2 columns of this table. And that's actually all I'm going to say about this, because next month, our March office hours will be about disease annotations at the Alliance. So our presenters will be talking much more about what's in the information that's indicated there. So below the table, there is a download button, and you can use that button to download all of the data that's in this table. So all of the alleles and variants for this particular gene. There is a link that says download all alleles and variants for all genes of the species. And if you click that, you go to the Alliance download site that gives you the data for all of the phenotypic alleles for your species of interest. And then between the two, there is a button that's labeled View Details, View Detailed Alleles and Variant Information. And if you click that button, it opens up a page showing detailed information about all of the alleles and variants associated with your gene of interest. So if we click that button, it takes us to this page, and you can see that at the top of this page, there is that same sequence viewer that we saw on the gene page. And then below that, there is a table that gives many more details about the variants in particular. So the first 7 columns of the table, this is the first 7 columns of the top of this table, and it gives records associated with variants are prioritized in this particular table because this table doesn't give additional information for alleles that don't have variants. So the first 7 columns are the same as the information on the gene page, which is basically all that is given for alleles in the category allele. And you'll notice that there are multiple rows for each allele or variant where there is a variant. And that's because the next 8 columns of the table give transcript-specific information about the variant. So you can see that this first column that's being shown here is labeled sequence features. So these are the transcripts for the LEPR gene in rat. And so these columns give information about, for instance, the location of the variant. In this case, the variant is located in exon 3 out of the 19 exons that exist in this transcript. And then also molecular consequences. So this variant is a frame shift. This variant down here is a missense variant. So information for transcripts from both NCBI and Ensemble are given where both are available. So you don't have to choose one or the other. It gives both. And then all of these variants have been submitted to Ensemble's variant effect predictor, which predicts the molecular consequences, so missense, frame shift, also the severity of the impact of the variant on the function of the corresponding protein based on the molecular consequences. So VEP impact for a missense variant is considered to be moderate. For a frame shift variant, the impact is high. And then where it's available, it will give the SIFT predictions of whether the variant is predicted to be deleterious or not. So in this case, this missense variant is predicted to be deleterious, and where it's available, there'll be a SIFT score. Where 2 or more genes overlap a variant position, like this one, the results are given for transcripts from both genes. So we have transcripts from both LEPR-OT and LEPR for this particular variant. A number of options are given at the top of the table for sorting the table. And in addition, the contents of the table can be filtered based on the data in any of the columns. So for columns where the number of options are limited, such as VEP impact, where there's only 4 options, then a list is provided where you can just choose which ones you want to see. So here I've chosen to only view results where the VEP impact is either high or moderate. where the filter allows free text, such as in the sequence feature column, the partial matches are accepted, and the search is not case sensitive. So I can put lowercase nm in and hit apply, and it will only show me the results where it's results for the refseq nm transcript. So going back to the allele invariant section of the gene page, I'm going to take us to the allele variant page. And the one easy way of getting to it is from this link in the first column of the alleles and variants summary table on the gene page. The first column of the details table also is a link to that allele or variant page for the allele or variant that you select. So if I choose LEPR fatty allele, LEPR FA, then it takes me to the allele page for that allele. And on that summary section that gives information about the alleles, such as a link to the gene that has been altered. So an easy way to get back to the gene page, a description of the allele, and then links to additional information in any associated research articles. Moving down the page, the genomic variant information section will give you information about the genomic variant. And in this case, there's only one. If there's more than one variant, then it will give a section like this for each of the variants that is associated with the alleles. So here it gives you the HGVS nomenclature as the symbol. It gives you a variant type. It's a point mutation. It overlaps the LEPR gene. It gives you a genomic location and then the nucleotide change. So in this case, it's an A to a C. It gives you the most severe consequence, which is the consequence that we've seen in the other two tables that we looked at. And then it gives a variety of HGVS nomenclature. So the various HGVR genome nomenclature, transcript nomenclature, and protein nomenclature are all given. And then at the bottom of this section, you'll see a link that says all alleles with this variant. In this particular case, this variant is only associated with this one allele, but that's not always the case. Sometimes a variant might be associated with more than one allele. So in this case, we're looking at an allele for one of the KRAS alleles in mouse and looking at the variant section. And if I click this all alleles with this variant link, it will take me to a result that shows me 7 different alleles that carry the same variant. And so this particular allele that we were looking at here, plus there are 6 other ones that carry the same variant or are associated with the same variant. So going back to the rat LEPR FA allele page, we move down to the variant molecular consequences section. This section gives the transcript specific consequences that we've seen before. But this page also lists the codon and amino acid changes. So in this case, we have the codon for glutamine, and we see that the CAG has been changed to a CCG that codes for a proline. And so you see the codons and the amino acid changes. Also, it gives the position of the variant in this particular transcript that we're looking at details for. It gives a position of the variant in the coding sequence of that particular transcript, and also the position of the variant amino acid in the protein. Below that, there are 2 sections, one for phenotypes that are associated with this allele, and another for diseases that are associated with this allele. And I am also, again, not going to talk about these because they will be looked at in more detail in our next office hours presentation. So we've seen a lot of information about alleles and variants, and we've seen positions relative to transcripts of a gene. But to see the variants in their genomic context, we'll want to go to the Alliance JBrowse genome browser. So going back to here, we're back at the LEPR gene page in the alleles and variants section. And if we click on this link, it will take us to the Alliance JBrowse 2 instance for rat at the position of the LEPR gene. And if you follow the link from the alleles and variants section of the page, you'll be taken straight to the browser with the tracks for genes, phenotypic variants, and high throughput variants all automatically opened. So if we click that, we get a view that looks something like this. And I'm not going to go into this in great detail. There will be an office hours presentation in May that will talk about JBrowse 2 at the Alliance. So if you want more information, tune in May. But I will point out a few things. So here we have the genes track, the track for the variants that are associated with phenotypic alleles. So we see those two variants that we've seen before, and then a track for all of the high throughput variants that we have for this rat gene. And then there's this sidebar on the right that gives options for additional information and what types of tracks are available is dependent on the species. So these are the tracks that are available for rat. There might be some different tracks available for other species in the Alliance. So one of the tracks that I want to point out is the Alliance orthology track. So if I click on that, then I open a number of options of types of orthology. I'm going to choose the most stringent. So these are the orthologs for which we have the most information or the most data to predict that a particular gene is an ortholog of the gene that I'm interested in. So here I've selected all of them, and you can see that for LEPR, there are orthologs in all of the vertebrate species, but not for worm or fly. And I'm not showing it here, but also not for yeast. But there are other genes in the area that have orthologs in all of the species, including the non-vertebrate species. So you can see the rat variants in the context of its gene and also orthologs in multiple species. So another nice thing that you can see in JBrowse 2 is you can see these multiple genome alignments. No, not alignments. Linear genome views in the same window. So here I'm showing the rat gene and phenotypic variant tracks, and also in parallel, the human LEPR gene and the ClinVar variants that are in this area. So this parallel view allows you to see variants in their genomic context across different species. So finally, I would like to give you a sneak peek at the proteins alignments and variants inspector tool that we are working on. This is a tool that is currently under development at the Alliance. So the idea is to be able to choose a focus gene. So here I've chosen human LEPR, and then choose orthologs in any or all of the Alliance species. So here I'm saying I want to see the human gene and the rat LEPR gene, and be able to see the variants actually embedded in the proteins with the proteins aligned. So the tool is designed to take the variant to make the change in the transcript and then to translate the altered sequence to give you an altered protein. And if you do that, then it will give you a view that will look something like this. This is not the final view, but it should look something like this. So here we have the human wild type, the human with one of the ClinVar variants embedded, the rat wild type, and then the two rat variants that we have been looking at, the missense mutation and the frameshift mutation. And here we're showing the region where the variant, the frameshift mutation variant is. And here you can see that the sequence is a histidine in the human, an alanine in the rat, but this has been changed by a one nucleotide insertion, changes that to a leucine, and then shifts the frame so the sequence is completely different after that, and it results in a premature stop about 50 amino acids down the line. So in this alignment, I'm showing the location of this ClinVar variant. So here, all of the sequences have a tyrosine except for this, where this ClinVar variant is, and that's a serine. And so you can see the variant embedded in the protein sequence and be able to see where the alignments give you information about where that variant is. And just to show you that there will be a number of different ways of coloring things, these two alignments are colored according to the conservation. And then this last one shows the Clustal-2 coloring scheme that is based on the properties of the amino acids. So it gives you some information about the properties of the amino acids. So that's just a quick sneak peek of the PAVI tool, and stay tuned. That will hopefully be coming soon. And with that, I would like to say thank you. I will take some questions, and also I'd like to give you some information about our next webinar. So I have the URL here to register for upcoming webinars and watch recordings of our previous office hour webinars in the event calendar. And stay tuned next month for disease annotations at the Alliance. Thanks. I'll take questions.
Thank you, Jennifer. So I believe people can unmute themselves to ask a question, or you can put something in the chat if you have a question. We'll open it up. Jennifer, how often are the sequence variant data updated in the Alliance?
The sequence data are the variants and alleles are updated with every release that we have. So that's approximately every 6 to 8 weeks.
And those are available for download too, bulk download. Is that correct?
Yes, on the downloads page. So actually, I can show it here. If you go to data and tools, the first option under data and tools is downloads. And so that takes you to a page where you can download the alleles, or that have the information about the variants is in that file, or you can download just the information about the phenotypic variants themselves.
Great. Any other questions from the participants if you raise your hand or unmute yourself or put it in the chat. All right. Well, Jennifer, thank you very much. Thank you, everybody, for coming. And hope to see you all next month at our next office hours presentation.
Thank you.
Thank you.
Thank you.